Alison, welcome to this discussion session that we have every month on cell and gene therapies, and we share information. In this, so I met Alison at the ASCGT meeting in Boston a couple of weeks ago, maybe last month, and I was enormously impressed at the presentation that she gave, of course, about Angelman's disease in multiple ways. One, obviously, the disease itself and the focus on the disease and the patient advocacy aspect of it. The second is, I think, the speed at which you've been able to do collaborations and the level of collaborations that you've been able to achieve in this short period of time. And then finally, the actual presentation itself, just a very, very engaging presentation. Thank you so much for accepting our invitation to come.
Oh, my pleasure. Thank you for inviting me. And I'm going to share with you our experience, my lived experience as a parent, and take you through our journey of drug development for a rare monogenic neurodevelopmental disorder called Angelman syndrome. This is a different journey than probably any of you have been on. And it's because the purpose is probably very different. And it's not that we don't all want to come to the point and the North Star of the same purpose. But when you live with a rare disease, things are just a lot different. And your urgency, your sense of urgency is certainly very different. And so I'm going to really try to show you our bench to bedside approach for rare disease and how that has been shaped by the lived patient experience. Okay. And I'd like to start with just, one really brief statement and something that anybody who is a parent or wants to become a parent should really think about. And everyone always asks me, how did you do this? And why did you do this? And I think really the question should be, what wouldn't you do for your child? Not what would you do for your child? Because I think that that's really what frames the path parents take, is that it always comes down to why not. So in introduction, I am the Chief Science Officer at the Foundation for Angelman Syndrome Therapeutics, also called FAST. I'm the co-director of a pre-competitive consortium called the ABOM, or the Angelman Syndrome Biomarker and Outcome Measure Consortium. I co-direct really a brain trust called InSync, which is the International Angelman Syndrome Research Council. And this brain trust is an international group of people from neuroscience all the way to business development folks in industry, drug developers, regulators, clinicians, endpoint developers. It really is a team of people, most of which are not working on Angelman syndrome. We brought together a team of individuals to help support the gap analysis in Angelman syndrome. Because when you start progressing drug development, you really do start to realize that you no longer know what you don't know. And when you get to the point where you think you've solved everything and you think you've touched on all of the gaps, that's when it's time to find new advisors, because it means that you don't have the right people in the room. And so we created this council of people that have done different things in different ways for different diseases to look at Angelman in a new light in order to be able to help us really understand the gaps we have in a different way so that we can ensure that we're doing the right thing from a drug development ecosystem perspective. I also serve as the Chief Development Officer of a company called AS² Bio, which is the Angelman Syndrome Drug Accelerator that was launched out of the foundation. And I'm responsible for all of the non-clinical activities from declaring the human clinical candidate all the way to filing the IND, and then clinical development. So all of the clinical trial designs and strategy behind the clinical trial. I'm a strategic advisor to other neurodevelopmental disorder gene therapy companies, one of which is called Mozzie Therapeutics, and I help to advance their non-clinical and clinical strategy. And the job that I actually have most of the time is that I'm a veterinarian. I'm a veterinary internist and an interventional radiologist. I actually trained at Penn. I lived in Philadelphia for 15 years. And I basically develop and design medical devices for animals with naturally occurring diseases in which there's a huge clinical and unmet need in animals. And then the translation of those devices is to the pediatric population, where medical devices are generally not developed for children. they're developed for adults and they're squeezed into children in some ways inappropriately. And so the devices we make for animals has really nice translation to the pediatric population. But the most important job that I have, aside from all of these, is that I'm the mother to 3 little girls. And my middle daughter, Quincy, she lives with Angelman syndrome. And so I'm going to start off with a video of what it's like a day in our life. I think it's important to understand the life of a parent with rare disease. And this is just a very brief video, but it's a story about Quincy and about our family. And I'm going to then take you through the patient journey, the clinician journey, what it's like to be part of an enormous community. Angelman affects 1 in 15,000 people. That's 500,000 people in the world potentially impacted by Angelman syndrome, in which there's not a single approved therapy for it. Then when we started multiple companies, there's a totally new journey that I had to go on, which is the industry journey. And that's very different than the academic patient and community journey. And then really what it comes down to is meeting that North Star of bringing transformative therapies to patients is the holistic journey. What does it take to bring all parts of this to patients and families? And that really requires bringing every aspect of these different journeys that we're on as a community and as a patient advocacy group to bring therapies to patients in need. And so hopefully I can share each aspect of this with you today. So let's start with just a really brief video, just so you can get to know Quincy. When you meet her, the first thing you will notice is her smile and her laugh. You'll also notice that she can't talk with her mouth. She talks with an iPad.
I like them.
Quincy has so many strengths and so many challenges. If I ask her to say hi, she opens her mouth and says, huh. She has Angelman syndrome. It is a deletion of one of her genes. I had a normal pregnancy with Quincy, but we started noticing things about her that were a little bit different. She wasn't engaging like a typical child. We were worried something is seriously wrong with her. So we asked if we can see a neurologist. The first words out of his mouth were, Miss Berent, I have catastrophic news for you. What she has been diagnosed with is Angelman syndrome. What stuck with me the most was not walking, not talking, and having severe seizures. Under treatments, it said no treatments available. My head just started spinning. All of the things you dream about for your beautiful newborn little girl, just taken away in a minute. And it was at that moment that I knew that I needed to educate myself and I needed to learn how to fix this, because mommy and daddy fix everything. Bad news either paralyzes you or it motivates you. And for me, it completely motivated me. By trying to create a therapeutic for Angelman syndrome. The gene that Quincy is missing is actually present in all of us, including her, but it's silent. And all we would need to do is unsilence that copy of that gene. And that would be a mechanism in which we can actually cure this disorder. 2 weeks after she was diagnosed, a paper came out, and it was in adult mice with Angelman syndrome. And they gave these mice a drug, and it reversed many of the symptoms that the mice had. And I realized that if they could do it in mice, why can't they do it in humans? How can we get this to my daughter? In order to get this from mice to humans, it requires a tremendous amount of work. I honestly felt in my heart, there's a treatment for this, but I didn't realize that we would have to discover it. Now, after all of this, the trial is open for enrollment, and I may be able to enroll my daughter in this study. This is a miracle that I can't believe I'm a part of. My hope is that this is transformative. I want to see her life improve, but I'm mostly thinking about who she is and who she might be. I just want her to say hi. That's what I want. So when you get a phone call and you pick up the phone and you hear on the other end, I have catastrophic news for you. Unfortunately, those are really the only words that stick with you. And you really don't hear the rest of what anybody says after that. When you're on a journey to figure out why your child is not developing like their typical sibling, you keep expecting they're going to catch up. And hearing those words really changes everything. And it's the before those words and the after those words. And that's how your life becomes defined. Then I hear she has Angelman syndrome. And all I asked is, how do you spell that? I had no idea what this man just said. And I did what every parent should never do. I Googled it. And I opened the first thing I saw, which was Wikipedia. And I saw all the things that Quincy will never be able to do. Never walk, never talk, never live an independent life, live a life of seizures, sleep 2 to 3 hours a night, never speak a single word in her life, and never live without adult supervision. And then I thought, well, what will she be able to do? What is her prognosis for survival? And how do I advocate to give her the best life that she's capable of? And it was very clear that I needed to advocate for Quincy. And I needed to learn everything I could about this disease if I was ever going to change the trajectory of what I was reading. And that was incredibly clear. And then I fell to the floor, to be honest. I was a very well-accomplished veterinarian who was exactly where I wanted to be. I was a professor. I had published my first textbook. I had published over 100 journal articles in PubMed, in high-impact journals. I was training interns and residents and students, and I was exactly where I wanted to be in my career. And it was only just starting. I was in my 30s. I didn't want to do this. I didn't sign up for this. I had no idea this was going to happen. This was a random event. And this is nothing that I ever experienced in my life. And this was not in the plan. And I fell to the floor because this is not the life that I wanted to live. And then the guilt. What kind of mother am I? How are we going to handle this like a family? How do we navigate a world of disability we've never been a part of? How do we manage a life of therapy for her and this full-time job that I'm not willing to give up? And all I could think about was there had to be a solution to this problem. I just didn't realize we might have to create it. Knowledge is power, and things are either possible or impossible, and there's no in between. And everybody told me it was impossible to cure a single gene disorder. But I couldn't find any evidence to support that this was not possible. There was just no data that anyone had tried. And so all I could think about was, well, if they haven't tried, maybe we should. And that's the path I took. I needed to be brave and I needed to be smart. And I called every senior author I could on every publication that I read, and most of them called me back. I attended every scientific conference I could, and what was so incredibly clear to me was a lot of work had been done on Angelman syndrome. Angelman syndrome is a very unique genetic disorder. It's an imprinted gene, which I'll explain. And the more I learned about it, the more I realized that there was so much potential to actually replace or unsilence this gene that only affects neurons of the brain. Why haven't we done it yet? And the good news is we have. We've actually rescued the phenotype in the adult mouse model of Angelman syndrome multiple times as of 2015 when she was diagnosed. Gene therapy, ASOs, multiple different strategies. The mouse was rescued. But what was so incredibly clear was that it was a great decade to be a mouse with Angelman syndrome. It was not the right decade to be Quincy with Angelman syndrome. And if there was anything I could do to change that, it was to help advance the translational science outside of an academic laboratory and into a clinical laboratory, into humans. And translational science is often really outside of the wheelhouse of most of the most brilliant minds who are developing these therapies. These individuals are working in academic laboratories, answering really interesting biologically driven questions. But the biologically driven question of how do we get this to a human is not the question that's usually asked. And because of that, more and more questions are asked based on the scientific discoveries that are had. But no one's realizing that when is enough to move to human, and when can the lab keep asking the really interesting questions. Who's going to advance it while the questions keep getting asked? And when it just stays in that lab, it doesn't move forward. It's a hamster wheel. And we were on a hamster wheel that was not helping Quincy, but it was helping mice. And if I could do anything with my training, it was to take the clinical candidates from these academic labs and get them through IND enabling studies and bring them to human patients. How do I connect the clinicians and the neuroscientists? And we often wait for a pharmaceutical company to come in and to rescue us because that's their job. But no one builds the bridge from mouse to human like a clinician. And if I could be a research scientist and a clinician, maybe I could help the team do that. And so that's the path that I took. And I realized very quickly we couldn't wait for a treatment, but we had to shepherd one, and I had to behave like a clinician. And so I understood everything I could about Angelman. It's a monogenic, non-degenerative, random neurologic disorder. The maternal and the paternal allele are sitting here on the left on chromosome 15. And if we look at the schematic of that, it is an imprinted gene. So there's this long non-coding RNA that's sitting over the UBE3A antisense transcript only on the paternal allele, and it's only imprinted and paternally silenced in neurons, where the rest of the cells of the body are biallelically expressing the UBE3A gene. In all of us, the maternal allele is expressed on the UBE3A gene, but the paternal allele is silenced by this long non-coding RNA. But in Quincy, the maternal allele is deleted. So she does not have a maternal expression, and she does not have paternal expression. You and I also don't have paternal expression, but we do have maternal expression. So if we can unsilence this long non-coding RNA and express the paternal allele, she would have the single copy that she needs in neurons, just like you and I. That can't be that hard, or so I thought. The impact of that single gene disorder, of that maternal mutation or deletion, results in, like I said, a universal lack of speech, kids that sleep 2 or 3 hours a night, that have debilitating seizures, often can't walk, and if they walk, they have significant ataxia, so they hurt themselves and fall a lot. They have impulsive and self-directed behaviors. They're unable to live independently, and there's a huge clinical unmet need. But we can't forget the impact it has on families. The inability to maintain employment, the complete loss of sleep when you're sleeping 2 or 3 hours a night, not just in infancy, but until death. These kids don't sleep, which means parents don't sleep. And it's very hard to maintain a social life, a relationship, a marriage, and a job when you are sleep deprived for your entire life, not just on maternity leave. The anxiety, depression, and stress, the incredible fatigue, and the excessive costs can be incredibly traumatic to families, if you can only imagine. And I read this paper soon after her diagnosis on somatic mosaicism in Angelman syndrome. And what this said to me was that for individuals, which is about 3% of the population, have 1 to 5% of maternal UBE3 expression because it didn't happen at the level of the egg, but it happened after fertilization. So they have mosaicism. And they have 95 to 99% of the maternal allele that is not expressing, with 1 to 5% expression. Those kids don't have seizures, walk, and have some speech. That's a different child than I live with. I would do anything in the world to give her 1 to 5% expression and make her a mosaic. If kids have 20% expression, they're fully ambulatory and can speak in sentences. And if they have 40% expression or more, they're asymptomatic and they're considered neurotypical. So what does this mean to me? This means to me a little bit goes a long way. The enemy of good is better. If we can get to 1 to 5% with a CSF-delivered AAV9 gene therapy, that could transform the lives of patients. But everybody's waiting for perfection, and everybody's waiting for 80 to 90% of neurons to be able to be transduced to say that we could do a gene therapy in the brain of people. In Angelman syndrome, you don't need that, because transformation and improvement in life is what matters, and we can get there with as little as 1 to 5%, similar to Becker's for Duchenne muscular dystrophy. So if we know that, we need to start acting now and start iterating on the technology that's available and known to be safe. And we need to try to improve these kids' lives. And so then I took a community journey. This journey started in October 2015. I joined the board of directors right after Quincy's diagnosis at the Foundation for Angelman Syndrome Therapeutics. And I soon in 2016 became their chief science officer. I got to meet these amazing consortium of scientists that were working in every therapeutic modality in order to try to either unsilence or replace the UBE3A gene. And they were trying to scientifically de-risk every approach in the multiple animal models that we had and in the cell lines that we had made. And it was incredible to work alongside all of these individuals. But what was also clear is that we didn't have a roadmap of how are we going to get to human candidate, not just mouse candidate. How are we going to get to first in human, not just a human candidate? So I was tasked when I joined the foundation to create this roadmap of drug discovery and drug development. And I did that. And we needed a plan and we needed an ask financially of what is it going to cost. At the time, the foundation was doing very well, but had never raised more than about $400,000 a year, which in a nonprofit rare disease community feels enormous. But I'm going to be honest with you. I'm a snob from New York City. I can raise $5 million in 30 minutes for dogs and cats. I work in the largest not-for-profit animal hospital in the world that raises about $20 to $50 million a year. How are we raising $400,000 a year for these children? We are not asking the right people. We do not have the right presentation, and we do not have the right understanding of how to ask for money. And so we had to change the way we were asking and what we were asking for, which was not just to families, but had to be with impact donors and philanthropists. And so we decided to change the way we were asking. And that's when we had the largest influx of funds to Angelman syndrome. 6 weeks after we created a roadmap on the back of a napkin with a colored pencil, we asked for $5.8 million for a 24-month plan to get to go and no-go decisions on 6 therapeutic platforms for this one single disease. And we raised it in 6 weeks. And it changed everything. We committed to do this in 24 months to get to human clinical candidate, not to human, but to have a clinical candidate to advance through IND to first in human. And everything at that point changed. And what the roadmap looked like was we needed a strategy to replace the gene using AAV gene replacement therapy or hematopoietic stem cell autologous ex vivo gene replacement therapy or enzyme replacement therapy. We can unsilence the long non-coding RNA with antisense oligonucleotides, with CRISPR gene editing, with zinc fingers, or with miRNA. We can think about the downstream effect of the synaptopathy related to the UBE3A loss with small molecules that might impact synaptic function. And while doing that, we needed to have the right models, the right cell lines, the right endpoints and biomarkers, the right policy and regulatory strategy, and we had to understand how are we going to design a clinical trial in which we can measure meaningful and transformative change over time based on the natural history of the disease. And we had to further support the natural history studies that were ongoing. And this was the roadmap of how we were going to develop drugs for Angelman syndrome. And you can look at that in many different ways. The first gene therapy that showed profound rescue was a hematopoietic stem cell autologous ex vivo gene therapy. And as I showed you the video earlier, here's an Angelman mouse model that looks a lot like Quincy on a treadmill. And this is after hematopoietic stem cell gene therapy. A single mouse, visibly different, treated as an adult, not as a newborn. So what does this tell me? There's a prospect for benefit. There's something that could help these children, regardless of their age. This is the CRISPR gene editor, unsilencing the paternal allele in adult mice. Wild type on the left, AS untreated in the middle, and AS gene edited on the right at 8 to 9 weeks of age. So this is well into adulthood. And what you can see is that the Angelman mouse has been fully rescued. And if you want to look at that in larger cohorts across different ages with different therapeutic modalities, what you could see is that we had this Cray-Lox inducible model that had been created where you can induce UBE3A expression over a time course from prenatal through adulthood, from birth, juvenile, adolescent, and adult. And what you can see is this study told us the later you induce, the less rescue you get, which would make sense dogmatically from a neuroplasticity perspective. Except, what did I just tell you? Well, when you look at gene therapy, HSC, ASOs, CRISPRs, or zinc fingers, regardless of age, whether you're an adult, you're at birth, adult or juvenile, and if you look at the CRISPR program here, adult, juvenile, and birth, everything is equally rescued. So an inducible model with tamoxifen that's neurotoxic isn't helping us say what's going to happen with actually a transformative therapeutic. So we have to think about the science in a way that's so much less dogmatic and more translational on what therapies are actually going to be in human brains. It's not going to be an inducible tamoxifen model. And so maybe the tamoxifen model is not the right model for what's going to tell us what's going to happen in humans. I don't know. The only experiment that's going to answer this question is the human experiment, the greatest experiment of all. So we can't get stuck in animals when the data is contradicting, and we have every reason to believe optimistically that humans can benefit from genetic therapies. We prepared for clinical trials in the greatest possible way. This is a 20-year natural history study funded by the NIH, the FDA, our ABOM consortium, and then we moved to Europe and Latin America and the UK. And what you can see in this 20-year natural history study in over 600 patients, that kids with Angelman syndrome after age 4 are flat on all of the neurodevelopmental trajectories, the Bailey, the Vineland, they're completely flat. They completely poorly perform compared to their neurotypically developing peers. The red is a kid like Quincy with a deletion, totally flat. They don't decline. So we're not fighting against loss. We're not fighting against slowing progression of disease. All we're trying to do is improve trajectory of disease because we have no loss. A kid at the age of 16 looks exactly as a kid at the age of 4. They basically make gains in some areas until about age 4, and then they're totally flat for the rest of their life. And so that's what we're fighting against. So we created this consortium called the ABOM, where we really thought through endpoints and biomarkers that were measurable and meaningful to individuals living with Angelman syndrome, and thought about all of the ways in which we can measure those endpoints in communication, speech, fine motor, gross motor, activities of daily living, quality of life, cognition, sleep, seizures, challenging behaviors, and biomarkers. All of the things that matter to families based off of a disease concept model and a patient-focused drug development meeting. And we looked at all of the ways we can measure that, all of the ways that were validated by regulators, that were used in the natural history studies, that we could say can measure change in our kids over time. And we looked at so many different measures and ultimately spent $5 million to come up with a master protocol of what we understand the minimal meaningful change would be with certain measures, capturing every one of these concepts that matter to families and patients. And that is hugely important in drug development. And so now we have a master protocol that any sponsor can use. We share everything freely if it's funded by the foundation. And now we can truly compare drugs that head to head with one another so that we can call drugs that don't work and we can accept drugs that do. But we don't want a bad endpoint to fail a good drug or a bad endpoint to allow a bad drug to be approved. We need to make sure that good drugs help patients get approved. And that's what our goal is as the ABOM. And then in 2017, remember, we funded 6 therapeutic platforms to get to human clinical candidate. 6 months later, we found in human cell lines that we can increase the expression of paternal UBE3A while we knock down expression of the long non-coding RNA. That ultimately resulted in mice of improving phenotype in the adult mouse and in their brain turning on the UBE3A UBE3 gene by knocking down the UBE3A long non-coding RNA antisense transcript. So now we have prospect for benefit in adult mice that a human ASO could benefit a human patient. And this is where it all started. we took the license to this technology and we launched a company called Genetics Biotherapeutics. I was the co-founder of this company that was supported fully by the foundation initially. And then we brought in angel investors to advance through IND enabling studies. We got through those IND enabling studies, and I started my first journey in industry. We launched Genetics in 2017, where we took a nonprofit into a for-profit, and we got through toxicology, pharmacology, pharmacokinetics, regulatory, clinical, manufacturing, all of the hurdles that we needed, backed by a very small team of consultants and a very small team of full-time employees. We got orphan drug designation, rare pediatric designation, fast track designation. And we got to human clinical candidate and filed our IND in essentially record time. In about 15 months, we filed our IND and dosed our first patient 19 months later. And because we needed to then raise more money for the clinical trial, we couldn't do that on the backs of our community that just put all this money into getting to human candidate and then all of this money into getting through IND enabling studies. So that was the time that we actually decided to partner the program with a company called Ultragenyx Pharmaceuticals. Ultragenyx partnered the program, paid for the phase 1/2. We sponsored the program, and they acquired us after positive phase 1/2 clinical data. And that acquisition infused the foundation with funds and allowed us to create an accelerator to move the other programs and the other clinical candidates forward toward IND enabling studies and first in human. And so this is the Ultragenyx data. And right after Ultragenyx acquired us, Ionis and Roche launched their Angelman syndrome ASO clinical trials. Competition breeds competition, whether we like it or not. And I love it because it's the best thing for my daughter is for there to be multiple shots on goal. I don't care who wins, as long as somebody does. And that should be my daughter and the other 500,000 kids living with Angelman syndrome. And so what you can see in this data is that this is the Bailey that I showed you earlier here in this second pane on natural history study and communication. So receptive language. This is the flat Bailey that I showed you earlier in those red and blue plots. This is that compared to GTX 102 ASO on treatment over 1 year, over 2 years. And what you can see is that we've blown the natural history out of the water. Similarly, in cognition, blown the natural history, which is flat and doesn't change over time, blown it out of the water. And so these are individuals aged 4 to 17 years old. Ionis showed exactly the same thing in expressive language compared to natural history, in receptive language compared to natural history, and in cognition compared to natural history. And so this is very, very exciting data showing that we can improve lives. If you notice here, we have AAV gene therapy, HSC, ASO, and CRISPR. Let me bring you back to that. This was the ASO. The ASO showed the worst rescue in all of the mouse models. What about a gene therapy? What about a CRISPR? What about HSC? It looks far better than the ASO. Now, if we believe anything in mice at all, I have a lot of excitement about a single treatment therapy that can get to the brain, get to more of the brain, and last for a longer period of time in which the ability to learn is magnified. And so the accelerator allowed us to further these 3 programs, which we were funding at the foundation level. One thing I want to also show you is that, remember that this heat map here? Well, look at the heat map in people. Being able to see a child respond to their name, follow 1 and 2 step directions, communicate wants and needs, have a first word, have multiple words, have decreased impulsivity and maladaptive behaviors, have less awakenings in the middle of the night, walk upstairs, decrease the number of falls. These are all things that matter. These are things we can't measure in a mouse, but these are things that we can measure in people. And this is the heat map in 4 to 17-year-olds with Angelman syndrome, like Quincy. And so this is where everything really changes. So when we think about drug development, there are 15,000 drugs in discovery for every one drug that gets approved. That can take anywhere from 10 years to 20 years and can cost us $50 to $250 million for one drug to be approved. How do we do this on the back of lemonade stands and golf tournaments? This is just not sustainable. And this is why we had to create a different model. And so where the foundation shows its strength is at the valley of death, where drug companies can cancel and pause programs because priorities change, where early proof of concept data is risky, where early discovery programs often get trumped to later clinical programs. If we have positive phase 1/2 data in humans, it's very unlikely a drug company is going to pause, shelve, or cancel that program. But if we have positive monkey data or mouse data, it's very likely a drug company is going to put that on a lower priority list. And so this is where the foundation has committed. We will fund our programs through phase 1/2 if we have to, in order to show what is best for humans, not what's best for mice. So when Quincy was diagnosed and I joined the foundation, this is where we were. There were 5 programs in development, almost all of which were in academia, and all of which were in very early stage discovery, 3 of which were funded by the foundation. After that $5.8 million infusion of funds, that went to 15 programs, 10 of which were funded by the foundation. 2 small molecules went into clinical trials. 2 ASOs were in IND-enabling studies. Then that moved to 34 programs. 3 ASOs in clinical trials, the 2 small molecules failed. IND-enabling studies with 2 additional programs funded by the foundation, and it goes on. Then in 2022, biotech took a turn for the worse. The stock market crashed for biotech. That further got extrapolated when our government changed regulatory predictability, I should say, and the biotech stock totally plummeted with news from our FDA. So that led what happens to us. Well, our 34 programs went down to 24 programs. But notice, none of the programs that failed or paused were those funded by the foundation. The foundation now has 31 programs in development, a gene therapy in phase 1, 3 ASOs in phase 3, 2 small molecules at the end of phase 2, with 2 additional programs in IND-enabling studies, all of which are funded by the foundation. And so this is where we are today. 17 fast-funded programs, 2 programs we funded in the clinic, 1 small molecule program we didn't fund, just failed their phase 1/2, and we're about to get into the clinic with our CRISPR gene editor and our hematopoietic stem cell gene therapy to follow. And so what I want to emphasize to you is that numerous potential therapeutic platforms have been invested in Angelman syndrome. Angelman syndrome is unique with many elegant gene-targeted opportunities. We have very promising clinical trial data to date, but we must take all shots on goal and ensure that we fill every gap, accelerate potential strategies, iterate on what we learn, and ensure that we find the best solution for our children, not the only solution for our children. Because the traditional drug development is certainly failing rare disease communities across the globe, but we absolutely will never let it fail this one. And so this is why we launched the accelerator called AS² Bio. We launched this accelerator so that nobody could stop us. We launched it because we understood if we take a venture philanthropy model, we can disrupt traditional drug development. We can do what's right to meet the North Star of patient needs, because those patient needs are our needs. And our priorities will never change. We will follow the science. We will advance programs with good science, and we will cancel programs when there's a very clear scientific no-go decision. And so we've created this bench-to-bedside roadmap, this model where we have this urgent mission to find transformative treatments for a global community. We have funded over $60 million in translational research in 30 academic labs with over 95 research grants. We've established 4 companies. We've launched 2 programs to first in human and are about to advance our next 2 programs to first in human. And we leverage this bench-side to bedside model with everything we do. And we leverage it with things other people do. We don't need to own the program. We just need to help people advance their programs. And so because of that, it gives us an opportunity to make sure that the patient's needs are met. And that's why we're here. And so with that bench-side to bedside model, we actually were able to leverage that through the Somatic Cell and Gene Editing Consortium, the SCGE, at the NIH. And we actually got the largest grant ever for the NIH, which is $40 million, to be able to advance our gene editing platform for Angelman syndrome and other indications behind it as a platform. And we got this money in 2023, and this is the CRISPR program that we're advancing. We then got a grant of $5.8 million from CIRM, the California Institute of Regenerative Medicine, in order to support our hematopoietic stem cell gene therapy in collaboration with Don Cohn at UCLA. And then we launched our Maverick study, which is our AAV gene therapy first in human CSF delivered gene therapy. And we're in that human clinical trial right now. And we've now been dosing patients for the last 6 months. And so really what you need to understand is that you can be platform agnostic and disease specific and take what you learn and apply it to other diseases. And that's a different path of drug development. And that's the path that we're taking for Angelman syndrome and other like neurodevelopmental disorders. This allowed us to get an ASO across the line, an AAV into the clinic. Our CRISPR and our HSC programs hopefully will be in the clinic in the coming year. And then other undisclosed programs that are in, close to follow in earlier stages of development. And so what I want to share with you and hopefully conclude is that drug development is hard. There's no question about it. I live it every day. The trials and tribulations of drug development are not for the weak. And I agree with every industry person who says that. But what I want to tell every single one of them is that living with rare disease is so much harder that we all need to put this into perspective. Who are we doing this for? I can tell you as a parent of a child who has a lot of challenges, her living with Angelman syndrome is dramatically harder than what I do every day in drug development. So we cannot stop until we meet the needs of these families and these children. But it's very hard to rely on other parties to make your disorder a priority when their priorities might change overnight. And I can't blame them. They are accountable to their board of directors, and they have to raise money based on the desires of their board. That doesn't mean that they're raising money on the desires of Quincy. And so this is a different type of drug development because it means different things. But we're all looking toward the same goal, because by helping patients, we will make money. And the return on investment for me is different than the return on investment for investors. But if we are successful, it will be the same, because the success of a child will be the success of an investor. And so FAST will continue to accelerate excellent science and make sure that we accelerate all shots on goal, that we adhere to our go and no-go decisions, and we don't push science that doesn't make sense. But we need to have common sense drug development. We need to work together to improve the efficiencies and serve as an example for others, because we've really shaved the fat off of drug development that happens in larger industry settings. And we really work toward and for the patients. So I hope what I'm able to share with you today is that things are either possible or impossible, like I said on my first slide, and there's no in between. But I think so far it looks possible, and we're going to keep going until we can confirm that it's possible. Because I believe anyone that told me the day of her diagnosis it was impossible to develop a gene therapy for her was 100% wrong. So this is my reason. This is my one reason out of 500,000. But we not only owe the 500,000 kids and adults in the world living with Angelman syndrome an opportunity, but what about the millions of others that are living with rare genetic conditions? Everything we're doing here could apply to them. We need to grow this at scale so that we can help the rest of them. Thanks.